Welcome to section 8.1, Mendel's experiments. We'll see how a careful set of pea plant crosses revealed simple, predictable rules for how traits pass from parents to offspring. The big idea? Traits aren't blended. They're passed as discrete units we now call genes. Our two goals for this section are first, to explain why Mendel's approach worked so well. His choice of pea plants, true breeding lines, careful controls, large sample sizes, and math-based analysis. And second, to describe what to expect from a simple single trait, a monohybrid cross, when one allele is dominant and the other is recessive. Specifically, the all-dominant F1 and the 3-to-1 ratio in the F2 generation. Gregor Mendel was an Austrian monk who loved careful observation and record keeping. Working with pea plants in a monastery garden, he tracked traits like flower color and seed shape across generations. His results challenged the 1800s idea of blending inheritance and instead pointed to inheritance by discrete units. His work wasn't widely recognized until around 1900, decades after he conducted his experiments. Why peas? Well, they self-fertilize, so they're easy to keep lines pure. They can also be cross-pollinated by hand, so Mendel could control parents. They grow fast and produce many offspring, and they have traits that are easy to see, like purple versus white flowers. Mendel also used true breeding lines, which consistently produce the same trait, giving him a reliable starting point for crosses. Here's the classic flower color cross. Mendel began by crossing true breeding purple pea plants with true breeding white pea plants. He called that the parental, or P generation. The offspring of that parental cross, called the first filial, or F1 generation, resulted in all offspring having violet flowers. Because the white color completely disappeared, Mendel called the purple color the dominant trait. Then he let the F1 plants self-pollinate to produce the second filial, or F2 generation. The F2 results were conducted hundreds of times, producing a ratio of approximately 3 violet pea plants for every 1 white pea plant. This consistent 3-to-1 pattern told him traits are carried by pairs of factors, which we now know as alleles, and that the recessive allele can hide in the F1 generation and then reappear in F2. Importantly, he counted lots of plants, so the ratios were statistically clear. Mendel chose traits that came in clear categories, discontinuous variation, like purple or white, not a blend. That made patterns obvious. Contrast this with continuous traits, like height, which vary along a range and are harder to sort into neat groups. Clear categories helped reveal the 3-to-1 ratio in F2. A dominant allele shows its effect if at least one copy is present. A recessive allele is only visible when 2 copies are present. In pea plants, purple flowers are dominant to white. Here's a vocabulary tip. A gene is the unit of heredity. Alleles are versions of that gene. Genotype is the allele combination, and phenotype is what you see. For the flower example, F1 plants are heterozygous and look purple. F2 includes both violet and white plants in a 3-to-1 ratio. Mendel's success came from smart design, true breeding lines, controlled crosses, big sample sizes, and math. His monohybrid crosses showed that traits are governed by alleles with dominant and recessive relationships. F1 shows the dominant trait, while F2 reveals a 3-to-1 ratio. These insights launched modern genetics.